Kia ora koutou everyone, and welcome to our first Women in Science event for 2022. I'm the Head of Research at the Neurological Foundation, and I'll be hosting our discussion tonight. I want to especially welcome anyone joining us from Invercargill. We're really sad we couldn't have the event in person there, and I know our panel was really looking forward to travelling to Invercargill. So hopefully we'll be able to do that at some stage in the near future. We'll keep some time at the end for questions from the audience. So if there's anything specific you'd like to ask our panelists, please type your questions in the Q&A box. So in New Zealand, neuroscience is full of talented women, and tonight we're very lucky to have three of them join us. Dr. Louise Bicknell is a senior lecturer at the University of Otago and is the co-deputy director of genetics Otago. She also co-chairs our personal awards committee that reviews all our scholarship and fellowship applications. And tonight she's joined by PhD students Megan Mulligan and Sankalita Ray. Thank you all so much for joining us tonight.
Thanks,
I'd like each of you to start by telling the audience a bit about yourself, how long you've been involved with research and what you're currently researching. So I'm going to put Louise on the spot first. Let's start with you, Louise.
Thanks, Sarah, and thanks everyone for joining us. As Sarah said, it's a real shame we couldn't be in Invercargill tonight, although Dunedin has got nice weather for a change. So I've been in research for about 12 years or so since finishing my PhD. And I'm interested in lots of things about biology, but mostly about the genetics and our DNA and how we can try and understand how our body develops and grows by studying our DNA. And so my lab, which Megan and Sankalita are part of, we focus on rare genetic disorders that particularly affect New Zealand children, focused around brain development. So we study syndromes such as intellectual disability or microcephaly, which is where your brain is not growing as expected, growing a bit small. So I've always been really interested in genetics, and it's been a slightly wandering path to get here, but really enjoying kind of what we do, and that we have quite a broad kind of different projects happening in the lab as the lab seems to get bigger and bigger and beyond my control.
Oh, I guess I'll introduce myself. So I'm, as Sarah said, I'm Megan, and I am currently just started my second year of my PhD. I guess I'm a baby researcher, as I only started a couple of years ago when I started my honours project, but absolutely love it. And so my project mainly focuses on looking at the molecular and genetic mechanisms of neurodevelopmental disorders in particular. So as Louise said, like children with microcephaly, so smaller heads, as well as some patients, they have intellectual disability, some have seizures. So there is quite a little bit of range, I guess, of neurological defects. And so what I aim to do is I aim to look through the DNA of these patients to try and find the mutations, or I guess the mistake that has gone wrong in these patients. And then from that, I try and figure out, I guess, kind of what the gene might be doing itself, or how the patient's mutation actually disrupts the gene. So quite rewarding.
Right, so I guess it's my turn now. My name is Sankalita. I am an assistant research fellow in Louise Bicknell lab. I am in a kind of a transition phase from a PhD student to an assistant research fellow. So my project in Louise's lab focuses, as Megan also mentioned about, Louise also mentioned about, we kind of focus on neurological developmental diseases. But my project is a bit more interesting because we are using zebrafish as our model to understand about the gene mutation causing the reduction in the brain size in the patient. It's a very cool project, and I guess with this project we will be able to understand more about the developmental functions of this gene. So yeah, it's very interesting and very cool project, and I'm happy to answer any zebrafish related fun facts if anyone wanna know. So yeah, that's me.
Sankalita, do you want to tell everyone what zebrafish are and why they make a really good model for these diseases?
Yeah, sure. So zebrafish are very tiny, tiny fishes. So they actually, the fun fact about zebrafish is I'm from India, and the origin of zebrafish is in the Ganges, actually, the river, which is in India. So that's very cool. I didn't know that. And when I started working with the zebrafish, I found like, wow, that's something new. And also the zebrafish, zebrafish has some conserved, how shall I put it, like conserved genetic information like human being, which actually makes it a very reliable model to understand about the human diseases. And this, I think zebrafish can regenerate its heart, which is another fun fact about zebrafish. It's very cool, eh? Like, imagine if we could regenerate our organs, and like especially heart, why not? And you get through a heartbreak, you regenerate your heart, right? So this is so then I think this is very cool. Yeah.
That does sound really exciting. For my next question, I'm going to start again with Megan and Sankalita, because you're both early on in your careers, and we may have students in the audience that would like to know how they could do a PhD in neuroscience. So starting with you, Megan, what has your research journey looked like so far? Why did you decide to become a researcher? Did you take a standard route?
Yep, so I feel like I did a standard. I left high school after year 13 and then went straight to university, and I did my Bachelor's of Science in genetics. And then I did an honours year after that, and then went straight into my PhD after that, which some people would call me insane, but there's people that do it. And so I guess I didn't necessarily think I was going to go into research to start with. I knew I liked science and I knew I was good at it, but I guess during school, I kind of kept my options open so that I could go any which way. And even in my first year at university, I had genetics and sort of that sort of field as my major. But I mean, I took, I guess, a wide range of papers so that if I did want to go another way, it was possible. So.
How about you, Sankalita?
My story is kind of a bit different. As I mentioned, I'm from India. So my bachelor's was in India, and that was in zoology, actually. So unlike here, zoology is a mother subject of biology, which combines immunology, molecular biology, biochemistry, all sorts of other papers. And then after that, I actually went to UK for my master's. So I did my biomedical science master's there. And I was stupid enough to do another master's in research. So I have 2 master's degrees actually. And then after that, I came to New Zealand for my PhD. Going back a bit further, I actually wanted to, at a certain age, when I was young. So I wanted to become a medical doctor by profession. And I actually got into that. But then I didn't enjoy that, because I wanted to know why things are happening, not just prescribing my patient a whole bunch of drugs without knowing what it's doing and how our system is working. So that actually got me into research. So I kind of followed that path and ended up here. So my PhD was not in neuroscience. My PhD was in cancer, actually. So I have a bit of wide background. So my research comes from to some extent. I mean, I worked for pregnancy complications for women. So my some project I worked which involved preeclampsia. So we were trying to find some sorts of answers for that. And then I did my PhD on prostate cancer. And now I am doing entirely different things in Louise's lab. So I feel like, to me, it's more the thirst of knowledge drives me. Like, I want to know why things are happening and how we can understand that. So that's kind of pretty much my journey.
Oh, thank you for that. We find that's one of the things that drives our clinicians into research as well, is that they're treating their patients and they really want to understand what's happening and if they could make things better and find better treatments. So
sorry,
really,
no offense, I should have said that. I mean, I didn't mean to offend anyone, but I mean, absolutely sorry.
So yeah, we love clinician researchers as well, because that's a really exciting field to be in. So Louise, we want to find out about your journey as well past the PhD. You've had quite an interesting, exciting journey doing lots of fun things. Do you want to tell everyone a bit about your journey and where you've been and what you've been doing?
Yeah, sure. Yeah. So I did my PhD here at Otago, and when I left, I thought, that's it, I've had enough of Dunedin, I'm never coming back, but here I am. And so I moved from the Edinburgh of the South to the Dunedin of the North. So I moved to Edinburgh in the UK to take up a postdoc, which is a pretty typical position that you do after you've got a PhD. And it's normally in a different lab, and it's a great chance to travel to another country. And I chose the UK because I spoke English, and my grandparents were British, and so I meant I could get a good visa so I could stay for ages as well, and managed to find a really good lab. Actually, during my PhD, I was going to a conference in New Orleans in the States, and my supervisor said, well, I know you want to work in the UK, you're kind of going halfway, just do another big flight and get to the UK and go and meet some labs that you might be interested in working with. And so I did at a few different spots around the UK, and then particularly kind of gelled well with this group in Edinburgh. And so that's luckily where I ended up about a year and a half later, and absolutely loved it. And I was there for almost 8 years. So about halfway through, I managed to get some funding to support myself. So rather than my boss paying me, I had external money to support myself and a technician to help me in the lab. And then when that funding was starting to dry up, I was kind of looking at moving back to New Zealand because it's home, and had some young children at the time and wanted them to grow up in New Zealand. And so we moved back to Dunedin. My husband's British, so first we did a wee tour to check that he'd be okay living here, which thankfully he approved. And I was really lucky to get a really good fellowship from the government, called a Rutherford Discovery Fellowship, to support me moving back and starting up my own independent research lab. And so that has been fantastic. It's been life-changing. It's a really good fellowship. It kind of opens a lot of doors for you. So I was really, really lucky and still quite humbled to have that. But it is 5 years of length, so it is limited. And as that 5 years got closer and closer to that end point, it starts to get more stressful. But last year I was lucky to get a new position in a different department as a senior lecturer. So that means that I still do the research, and that seems to keep me really busy, but also teaching into the undergraduate courses, and then do some service and engagement opportunities as well. And that job is permanent, so that's huge for a scientist to have a permanent job.
And a big congratulations to you for landing that role at the University of Otago, because I know how difficult they are to come by. So
yeah. It was very emotional to get that one.
One of the things I love about having Louise on our personal awards committee is that she's really enthusiastic about research and really passionate about helping others advance in their research. So I'll go back to you first again, Louise, with this question. What excites you most about your research? Every day it's different. Well, that's one thing that is exciting about doing research, is that no 2 days are the same, unless you have a lot of failed experiments, which hopefully they don't have. But what excites, it's always different. It could be an email that's come in overnight from an international collaborator about a new patient. It could be hopefully some fantastic data that like Megan or Sanjanas have generated that they want to show me, or just reading a new paper and getting ideas. I think that's what's exciting, is that there's no 2 days the same, and that also your motivation changes depending on what
project it is. So some projects are much more motivated by the people behind them and the families that we're trying to help. Other times it's just trying to understand, what's going on here, what's the biology, what are we trying to understand? And when we get a bit of insight into that, that's always exciting. And other times it's just like what is going on with DNA and just the real power for us to find these spelling mistakes that we talk about. Like what on earth, how is this happening? Why are we seeing so many kids with the same spelling mistakes? Just the power in there is really, really exciting. So it doesn't take much to motivate me, as you can tell. But yeah, it's always different. And we're really privileged to be able to have a job that has so much flexibility and, and imagination and scope.
How about you, Sankalita? What excites you most about your research?
I guess, as I mentioned earlier, I just want to know why things are happening. And I remember my paper during my undergrad, there was a paper on embryology. And I think at that point, when I read that, I was going through that paper, it just starts to fascinate me how from one cell, this whole thing is standing right now, like how and what level of, can you imagine how organized and orchestrated that program is to build up this from just one cell? So this kinds of question actually drives me crazy. So I think that's one of the reason that I wanted to be in research. I'm not saying that I'm gonna know or I'm gonna find all the answers, but at least I will find some answers. That way I can help the society to know that with that information, so yeah.
How about you, Megan?
I guess there's a couple of things that excite me a lot. One of the things that I've been very excited about since working on sort of brain development type things is actually being able to grow brain cells in a flask, in a dish. Like it's crazy that we have the resources now to be able to take brain cells to sort of mimic, what might be going on in our patients, or to better understand functions of things. I think that excites me a lot, even though I spend countless weekends in here trying to deal with them, and they often don't grow and they annoy me sometimes. I think it's very, very cool. And I think another thing is it's very exciting to find a mutation that's causing something in a patient. So just before Christmas, I found my first causative mutation, which I was very excited about. I came bounding into Louise's office, very, very stoked about it. So that excites me, because not only are you helping patients, you're helping the family, you're giving them some sort of answer, then you're also looking, okay, how has this been seen before? Is this kind of a normal, I guess not a normal thing, but is it common, or is this a total rare novel thing that we now have no idea about? And now that excites me to go, okay, so this gene does what is this doing? And then it just leads you down a huge rabbit hole of why, like Sankalita said, why is it happening?
So what is the time frame that the family would find out about this mutation, and is that something you're involved in, or do you pass that information on to a clinician or a genetic counsellor?
So I relay it to Louise, and then Louise then contacts the clinician, who often they ask for updates about the patient, which is when we often prioritize looking through their DNA a bit quicker. But sometimes, I'm looking through patient DNA at the moment where we haven't had an answer for quite a while. So we just keep resequencing it, or do bigger sequencing efforts to try and find that. I don't know if Louise has anything to add about that.
Yeah, no, you're right on there. So it can depend on what kind of answer we find. So we tend to find answers in about half the patients that we study. And sometimes they're really quick, because they kind of just pop out of the screen at you and it's a pretty obvious answer. Megan's ones were pretty, yep, that's definitely it. And it's because it's been studied before, and there's evidence already, pointing this as to being the answer. And so it just depends on how much evidence that for us to be able to be certain that, yep, that is the actual answer. And then we always check with the clinician to say, this is what we found, we think we're onto something here, what do you think? They're the experts on their patients more than us, so we always make sure that it can include, everything tees up. And it's important to remember that we work in a research environment, which is slightly different to a diagnostic environment. So what we always ask is that anything that we find, the diagnostic lab kind of repeats the test and confirms it, just to make absolutely sure that we're giving, the accurate information before the clinician gives it back to the family. So sometimes it's really quick, and those are really, they're great for the family, but often it means there's kind of a shorter research journey for those particular families and projects. Whereas if it's a gene that perhaps hasn't been associated with a disease before, then that's a much longer term answer, but slightly more exciting from a research point of view, because there's kind of more insights and more why to be understood.
Yeah, finding out something new.
Yeah.
And I think it is a very exciting time for neuroscience research. It feels that technology at the moment is advancing really, really quickly. And having been a neuroscientist myself, I might be a little biased, I suppose, but I also think that neuroscience is one of the most challenging areas of research. And what do you think, Louise? What are some of the challenges that you face in your work?
Yeah, that's a great question. I think from a research point of view, from a purely thinking about our DNA, is that everyone, when you look at different people, everyone's got tons of differences between the person that you might be sitting next to or you might be living with. And so a big challenge for us is to weed out the changes that we don't think are causing the disease that we might be studying. And there's some tools that can help with this, and these tools are really quickly developing. But at the same time, we really understand very little about all of our DNA. Our DNA is massive, and the bits that encode these genes that we talk about actually only make up about 2% of our genome. So there's a huge amount of DNA in our genome that we don't fully understand, and it's certainly coming along, but it does make it a challenge for us if we find a change that isn't in one of these genes or in these well understood areas. We can't do much more with it at this point because we don't understand what the consequences could be, or how big a deal is it that's changing. So I think that's, from a purely DNA centric kind of view, that's kind of the biggest challenge. And there's huge big groups around the world working on that, trying to understand them more, but it's a complicated question to generate all of that data.
And what about you, Megan, both in study and work, your work on your PhD, what do you find the most challenging?
I guess a big thing was getting used to having to be a bit more independent, especially for a PhD. I guess during honours and undergrad, your hands basically held. It's kind of like, we've found this, you can do this experiment and it will probably show you that this is happening. Whereas now I have to use my brain quite a bit more, which can be quite tiring, but very rewarding at the same time. So I think a big challenge here was the shift from being told what to do. And I think Louise would agree with me, I'm still shifting to there. But yeah, I think that's quite hard in itself.
Yeah, I definitely noticed doing a PhD is a huge time of growth in many ways.
Yeah, definitely.
What about you, Sankalita? You've obviously done a lot of different roles. What are some of the challenges that you come across in the research field?
I think for me, right at this point, is I just kind of crossed that PhD student phase. So, as a PhD student, no offense, Megan, but you just don't have your own time. Like,
you don't have a life. I can agree. We do not have a life. I mean, I'm still in the office.
So
to encourage a healthy work-life balance, I should point out.
But then now I'm working as a staff. So I remember me and Louise, we were having a chat one day about this. It takes a time to transit from that student phase to become a staff. Like, you are allowed to say no for certain things, like those kinds of thing. I feel like sometimes PhD students don't realize they are human being as well. So they have all the says, and they can say that, no, it's not okay. So I think this kinds of challenges, I kind of, I felt that's, yeah, it's challenging. But for me now, it's like that transition phase. I'm trying, Louise, to get into that phase that, yes, I'm no longer a student, I'm a professional. So I find it challenging. Yeah.
And obviously, over the past 2 years, the pandemic has hugely disrupted research in New Zealand and also international travel. And I know that international conferences usually play quite a big part in advancing research. You've also mentioned, Louise, you have a lot of international collaboration. So how important is that international collaboration to your work, and how has it been affected by COVID?
Yeah, yes, it's critical for our kind of work. You know, the patients we study are very likely to be the only patient in New Zealand with that alteration that we're finding, that spelling mistake. And so that means if we want to gather more genetic evidence to say, yes, this is definitely what's causing their condition, we need more patients, and that means looking outside of New Zealand. And so having international collaborators is critically important to that. So I have built up a really good network of different people I can email and ask who might be studying similar things in different countries, similar kind of features, and ask them if they've found anything. We also rely on, I call it a dating site for geneticists, where you can put in your favourite gene name, the gene that you are looking at, and other people around the world have done the same thing, and then you get matched, and you can swap email addresses, and you can start to find out a bit more about, you know, what their patient has got, to see if it might line up. And that's been really successful for us, and has, you know, led to lots of new collaborations as well. So it has been really important. And I think COVID has meant things like, not so much here in New Zealand, but to a certain extent as well, is that often, like, you know, genetics appointments or going to see a clinical geneticist might get deprioritised in the healthcare system. And we rely on those to identify patients that might be able to be involved in our research. We don't approach patients directly. And so obviously if those appointments aren't happening, we can't then get more patients involved in our research, and so that kind of slows it down. And also being kind of at the bottom of the world, shipping things is taking much longer. So we've tried to get skin cells from patients sent over from Europe to kind of look at things, but after tracking them for about 10 days through the US and all over the show, it should only take about 5 days, and 10 days time to push that the cells survive. So it's really kind of more risky to want to send those cells in the first place because they're so precious. So it's, yeah, kind of affected us in different ways. We've been really lucky. We're now starting to plan for what might be coming in the next couple of months. But we certainly have felt the effects.
Yeah, I think you have sort of escaped quite well in Dunedin from most of the lockdowns, but Sankalita, how has different restrictions affected your work?
In terms of my work, I actually, when I was finishing my PhD, right in the middle, I just recently handed in my thesis. So right in the middle, there was a lockdown. So it actually impacted my experiment a lot, because when we do an experiment, we don't think about that culturing those cells, or I work with cell lines, so culturing those cells, bringing them up from the liquid nitrogen, give them enough time to set up an experiment. These things also count. Although it seems like, oh, there was just a 6 weeks of lockdown or something like that, but actually that impacted, it's a domino effect, you know, everything gets pushed. And as Louise mentioned, that all the deliveries and all the ordering has been delayed and delayed and delayed. So that actually impacted my research quite a bit at that phase. So I had to kind of wrap it up within kind of in the middle of that pandemic and lockdown, and just handed my thesis.
So you didn't get to finish exactly all that you wanted to get done?
I couldn't the way I wanted to.
Yeah.
If that wouldn't have happened, some of the experiments, some of the data would have been nicer. Like, you know, that satisfaction. I'm still proud of my thesis and proud of myself that I was able to finish that during that, given that all situation. However, I feel like I personally would have been more happier. Like, yes, that's what I wanted, you know. So yes.
And how about you, Megan? I know in Auckland, a lot of the students have been pretty stressed, and are you worried at all about what the impact is going to be on your PhD?
I guess I haven't thought long term yet on the impact it might have overall on the PhD, but in terms of right now, yeah, as Sankalita said, experiments. I mean, I think the August lockdown, I was all set. We'd been waiting, I think it was about 4 weeks or 5 or 6 weeks for new cell media to come in. Finally got here, and then that night, I actually put my cells in the flask, did everything, and then 6 o'clock I had to go back in and throw them out. So yeah, it just delayed a lot of things, and then yeah, as well as the shipping and the ordering, things seem to get lost. They seem to forget about New Zealand in general. It's worse with the pandemic. So yeah, no, I'm not sure yet on the long term, but as I get closer to my 3rd year, I think it might be a bit of a rush to get all my stuff together and all my things in line.
Well done all of you for keeping going in these challenging times. And I think in research, it's really common to always be looking ahead about what you want to achieve and are you going to get there. But I just want to take a little bit of time tonight to celebrate what you have all achieved so far, especially in such challenging times. So Louise, what do you think are your most significant accomplishments so far?
Great question. I think personally, actually, managing to get a permanent job is huge, and that might sound trivial for those that aren't in this field, but they are so rare. And it's really stressful living, you know, often, so Sankalita is now experiencing this kind of unfortunately. If you're a researcher working in a lab and you're funded by grants, you might only have a 1-year contract, and so you're living kind of year to year. And so that really impacts your life and, you know, your kind of longer term plans. So having that stress kind of removed, I can feel the difference, you know, in me of just how that, that stress isn't there anymore. And I can think about longer term science projects that I want to do, knowing that I don't have to kind of bounce from grant to grant to kind of keep that going. So I think that's kind of been, in addition to all of the really, really cool science, and I probably couldn't pinpoint because all the different genes we study and things are exciting in their own ways. That's probably been the most significant accomplishment, is to stay employed. I set the bar low.
And how about you, Megan? So early on in your career, what are you really proud of accomplishing so far?
Yeah, I was trying to think about that. There wasn't, I guess, there's nothing, like I don't have any permanent job like Louise yet, but I think my biggest accomplishment was actually getting a scholarship for the PhD program. Right at the end of my honours year, I actually had a family bereavement and I kind of wanted to give up on life. So I was just like, I didn't want to write my thesis. But I did it, I managed to get the grades for the scholarship, and now I'm here. So very, very thankful for that.
Oh, well done. How about you, Sankalita?
I think for me, so far, my greatest achievement is surviving my PhD. So that's something I'm really proud of myself for that. I'm still waiting for my defense, but that's another story. But it's okay, it's fine. that's okay, we will get there. But I think that's my biggest achievement so far. In addition to that, I think I survived the pandemic, and I was blessed enough to have a job during this pandemic, where I know many people, many of my friends, they have lost their jobs because of the situation. And in a lab, like, I would say, yeah, I'm blessed enough to have a job in Louise's lab, honestly. So I think these are my greatest achievements so far.
Oh, that's fantastic. And that's, yeah, one of the reasons the foundation has really wanted to keep supporting people so that they have a job and they can stay in research and come out the other side of the pandemic still researching, which is really exciting. So I'm going to get you all to look a little bit further forward now, hopefully post-pandemic. What do you hope to achieve in 10 years' time? What are your long-term goals? This time we'll start at the other end with Megan.
Maybe. So it actually kind of scared me thinking about how old I feel like I'll be in 10 years. But anyway, Louise would probably laugh at me. But anyway, I think one of my main long-term goals is to actually leave New Zealand and experience research in an overseas lab. I imagine it's a totally different environment. And yeah, I think that's definitely my main goal, to go and be a postdoc overseas somewhere. Probably, hopefully not as cold as Dunedin, but yeah, other than that, yeah, I'm not too sure where I'll be in 10 years. I'm interested, but I'm not sure yet.
How about you, Sankalita?
I want to become a leader like Louise, because there are bosses, but I have come across many PIs during my career, but I think she inspires me. So if you ask me, do I see myself in academia in next 10 years? Maybe. But in next 10 years, I definitely want to get a permanent job and want to be a leader like Louise.
That's really nice. How do you respond to that, Louise? I'm sure you've got a lot of you've got a lot of big long-term goals, I'm sure.
Yeah, look, you know, it's funny, when I was doing like my PhD and then afterwards, I didn't really know if I wanted to stay in research long-term, just because there's a lot of ups and downs, and there's so much passion involved. You know, it's quite a personal thing really, and it's hard to kind of keep that separate when there's kind of, yeah, especially some of the lows, of how to kind of keep going after that. But now I can be a bit more kind of ambitious. So I have quite big goals really, and it's more focused around genetics and how the genetics service that we have in New Zealand and the linkages to research are really quite behind a lot of other countries. And that's not because of any lack of goodwill between clinicians and research. It's just kind of the clunky infrastructure and DHBs and all those sorts of things. So I really want to see us, you know, researchers work really alongside clinicians and really be quite closely connected, so that we really can maximise how we can help diagnostic rates and diagnoses in New Zealand, and give the information that the families need, you know, to be able to help inform their lifetime of, you know, what they can expect for their child, for example. So I think I've got a few different ideas on how we're going to do that, but yeah, that's the big goal. There's been lots happening overseas, and it just hasn't really taken off here. So hopefully I can help change that a bit.
Yeah, that's definitely something we're always looking at ways as a foundation we can support that clinician researcher linkage as well, because the results just come out so much faster that way. When you can go back and forward with the clinicians about what's actually happening in the patients. Yeah,
I think there's tons of opportunity. So, as I say, everyone wants to, but it's just getting the support to kind of start that happening.
So I'm just going to ask one last question, and then we'll start answering some of the questions from the audience. But what or who inspires you to keep going when things get challenging?
I can start if you like. So actually, often my inspiration comes from these two other people on the panel and the other members in the lab. So, you know, that they're so driven. Actually, I get a bit lonely in my office by myself sometimes, because they're so driven getting on with their science. And so it's really inspiring to see how passionate they are about their work, and how when I'm buried in admin, or, you know, things aren't going quite so great, their research is what brings me back to me, and this is why I really enjoy it so much. In addition to that, I have incredibly strong support from my family, both my husband and children, and then also my parents and sisters, to, you know, have a good rant occasionally when I need to have that release about when things aren't going so well. So yeah, tons of inspiration, and the families, of course. you know, that they're willing to be engaged and willing to engage with our research. We certainly don't take that as a given. Yeah.
How about you, Megan?
So I'm going to be cheesy as well and say Louise, because she definitely does inspire me to keep going, especially the amount of times I've gone into the office kind of like, oh, it didn't work, as well as I guess the rest of the lab as well. Yeah, they kind of keep me going when I think everything's failed and nothing's worked and it's the end of the world. So yeah, they definitely keep me going. And I guess myself to a certain extent. I mean, there's definitely certain times where I'm like, oh, I don't know how to fix this, or I've thought of many ways that I could try and fix it and nothing's working. But then you kind of step back a bit and kind of go, well, I'm really passionate about this. You know, I want to keep going forward. And especially, as Louise said earlier, when we are looking for mutations in patients, you can sit at the screen for hours scrolling through Excel sheets, finding absolutely nothing, just ready to give up. But then you kind of picture the family and just picture them, you know, wanting their diagnosis and being curious about what's going on with their child. So I think that inspires me to keep going.
How about you, Sankaleta?
I think I just answered before, or as I said earlier, that Louise inspires me a lot. In addition to that, I think it's my mom. She is kind of diva to me, and I really look up to her. Of course, my dad, my sister, and my best friend. I mean, you know, apart from your family, you need someone to go to and just like say everything to get that. So I think my best friend, he's kind of like, he has helped me to get through so many of my life phases. So yeah, these are the people inspires me a lot to, you know, wake up every day and get things done and feel about, and most importantly, be proud of it. Own your thing. Like, this is my thing. Whatever is happening, it can be good, it can be bad. And I kind of got that, probably that from my mom. She's like that. And now when I look up to Louise, and I can see that from her, like, it's okay. We will get it done. Like, so this kinds of, I'm blessed to have this kinds of people in my life who inspires me a lot.
Oh, that's really fantastic just to hear the support that you guys have got around yourselves. And I guess researchers are problem solvers, and you obviously can't get away from the problem. You won't leave it alone. You keep coming back to it until you find a solution. And that's why people donate to the foundation, to find these solutions. So thank you all for what you do. I'm going to mention one of the comments from the audience. And they say that living year to year sounds really difficult, and they want to champion you for sticking with it. And they ask, has that been a deterrent at all for other people that you've noticed about getting into research or staying with research?
Yes, I can probably answer that one. Yeah, absolutely. It is really difficult and really stressful. And now, so there's 5 people in my lab who I support their salaries, so as staff. And it's a really big stressor that I need to keep finding grants to keep these wonderful people employed and working on our really exciting projects. And that, you know, research takes time. So it is really stressful. And yes, it does cause people to decide to leave research because, you know, it becomes too much. And even just the short-term kind of contracts has an impact on kind of your CV, the kind of turnover of your research, how you can publish it, and then that kind of is a vicious cycle. If you don't publish your research enough, then it's harder to get more grants. So having the stability is a huge deal to be able to handle all of that. So it is really difficult. But then kind of on the flip side also, there's I think we'd need to probably bankrupt the government if everyone who wanted to stay in research ever could, because there just simply isn't enough money. So it is a really tricky kind of conversation. And I think it's really recognising that leaving research isn't a failure, because sometimes you're probably a bit crazy to stay in research, because these highs and lows. So actually there's so many more opportunities and the kind of skills you get from doing a PhD that, you know, the world's your oyster really.
So how hard is it to get research funding in New Zealand, and is it getting harder?
Yep, definitely. So it depends on the average kind of success rate for getting a grant that you might write. It varies. About the most generous and with kind of the easiest grants to get might be kind of 40% of the applications are successful, whereas kind of the hardest grants to get less than 10%, so up to about 9% of applications. And it's a huge amount of work to write a grant. So then to get a no email after all that effort, you know, is pretty disheartening. So it is getting harder and harder. And we notice that even at the foundation, so on the personal awards committee that I sit on, that, you know, we're getting more and more applications. So that's why donations and being able to hopefully in time increase how many fellowships and grants we can offer is would be amazing, because we have amazing researchers and just never enough money. So it is getting harder, and I don't know quite why. Maybe there's more researchers or just kind of other funding dries up, but it is, yeah, research is costing more money, of course. So yeah, it is getting harder and a bit more stressful.
And Megan, have you noticed if PhD students continue, or do some of them not finish their PhDs? What is the environment among students?
I'm not too sure necessarily about PhD students, but I know students in fourth year when I was doing my honours, I think there's only 2 of us that have gone down the PhD sort of academia path, whereas a lot of them, they kind of had a feeling, and they know that in academia the funding is hard and you do live off, you know, year to year. So I think there was probably 4 or 5 people that I know went into industry jobs where they could guarantee, you know, that they've got a contract for so many years. So I know it affected students in that way.
And Sankalita, obviously you've worked outside of New Zealand as well. What is the New Zealand environment compared to elsewhere that you've seen?
In terms of intellectuality or research, I would say New Zealand is no less than any other country, that's for sure. But in terms of funding, yes, we are struggling. That's true. That is, there is no sugarcoating onto that. Because I was in UK, I have seen the funding opportunity there. And not only that, in terms of industrial jobs, which Megan was mentioning, I just want to add on to that. I know all of my friends from UK who just finished, who finished their PhD, none of them, they are in academia anymore. They are in research, but not in academia. They love research, but they want more permanent positions, so they moved into industry. They are doing fantastic science there. They are even publishing from there, but they are not in academia. So I think this is a bit of stressful environment. Yes, I agree. But again, it's more what you want. And are you willing to take that risk, or are you willing to, kind of wait, or that uncertainty in your life? So yeah.
I think research often isn't somewhere you plan to end up, it's somewhere you find yourself ending up. So one of our audience is asking, how old were you when you decided to study and you decided to dedicate yourself to this job, and is it different from what you wanted to do when you were young? So maybe we'll start with our youngest. Well, Louise can try and remember back.
Crank.
I guess I always knew I wanted to go and study something further, as I think one of our main topics has been. My thirst for knowledge was always there, always the why. So yeah, I went straight into study from school. And what did I want to be? I think there was a couple of things. I did want to bake cupcakes for a living, but it was funny because I'm absolutely a horrendous baker. And I did want to be a doctor, but yeah, no, I grew out of that phase pretty quickly. So nothing, as a kid, nothing that I thought I might have done. Definitely didn't think I'd be sitting here growing brain cells and doing cool things like that.
I think, Sankalita, you mentioned you originally wanted to be a medical doctor as well. Was that when you were young as well?
Yes. I kind of grew passion for science after my board exam. So in India, it's board exams is like 10 and 10 plus 2. Those are your board exams. So during that time, I kind of grew the passion for science. And that, again, that reason was my mom. She's an amazing scientist, actually. So yeah, and then, but if I didn't go in this research career, I would have probably become an architecture. So that was, I love painting. I absolutely love painting. So that's one of the things. That's probably my alternative career, you know, if it doesn't pan out. But that's probably my alternative career that, yep, I can paint and I can sell that. So, yeah.
That sounds like a really good creative outlet as well for when things are difficult in the lab.
Yeah.
How about you, Louise? What were your childhood dreams and how old were you when you got into research?
Yeah, so I don't remember doing much science at school, and that's probably showing my age a wee bit. Like in primary school and things, certainly. I think I was just really curious to understand, and I liked maths and logic, and so I think that's why I kind of went down the sciences route. But back in my day, yeah, it felt like especially if you're a female and you like science, then you kind of did medicine. That seemed to be the go-to. And it was only when I came to university, which I was one of the first in the family to go, like my parents hadn't been to university or anything, so it was a bit of an unknown field. And when I came here, and I realised, yeah, I really don't want to do medicine. I want to be the one finding the things. I don't want to be just kind of delivering the news. And so, and during my first year, I got exposed to a bit of genetics and absolutely loved it, and anatomy. And so that's what I tried to do, those two things as a science degree, and then moved on to a PhD and just got hooked, really. And it just felt very natural to want to keep doing more and more research, and kind of just ended up here, yeah, part by accident and part by not having a plan B. I had no idea what else I would be doing if I wasn't doing this. And I think it's funny that even though I didn't want to do medicine, the kind of science that we do is kind of as close as to medicine as we can get almost, and that we're still helping patients and still working on with patient samples. So we're really close to it, but we're helping make those discoveries.
Yeah, that's really fantastic. And we've got a comment that I'll read to the three of you, which is not a question, but Nikita wants to say that she loves how supportive you are of each other and of others doing research, and that it's really great to see.
Oh, thank you, that's really kind, Nikita. But I think that's a really big part of it, right, guys, and that you very much are kind of this family. You spend your days often at the bench, you know, things not working, real science, things often don't work. It takes a while to get them working. And so you do really rely on that support network. And it's something I take really seriously when new people come into the lab, to make sure they do fit in and they're going to be part of that great support network to keep us all going. That, yeah, it's a really important part of it, actually, is that teamwork and camaraderie and having a bit of fun along the way.
So we've got a few thumbs up to one particular question. If you could meet one person, who would it be?
It's a really good question. I haven't got the foggiest, you know. I was like, oh, who's taking a spaceship to the moon? But actually, I don't really want to meet any of those males. For someone else.
Is there a key person in genetics that you'd like to meet or collaborate with?
I think, you know what, actually, it's really nice now that I think I've become experienced enough in leading my own lab that I've got the gumption by email that if there was someone I really wanted to get in touch with, I would, you know, and I wouldn't be too held back. And actually that's a lot of the reason we go to international conferences, which hopefully Megan and Sankalita will get to at some point, is to actually network and make those connections. And so when you do send an email saying, actually, could you help me with a project, or I'd love to do this with you, it makes it that much easier.
Megan and Sankalita, have either of you thought of someone that you'd just love to meet?
Does it have to be in science?
I don't think so.
Then I would definitely like to meet Jared Butler. Definitely. That's why I ask. Does it have to be in science?
What about you, Megan?
Not that I can think of that comes to my mind, but maybe I could put that as a long-term goal to think of somebody that in the next 10 years that I want to meet, because I'm not sure yet.
I don't know if you find this, but I found being involved in science and research, you're always meeting really fascinating people all the time.
Yeah, and everyone, as we've heard tonight, everyone's got such different stories and how they've come to be in research and what drives them and things. Absolutely.
So it's been fantastic talking to you tonight, and I feel like we could just keep doing questions forever and finding out more about science and research, and the three of you individually, and how you've made it to the stage that you're at. And I can see great things for you all in the future as well, and hopefully the foundation can keep being a part of your research journeys. So I just want to say a big thank you to you for joining us tonight, and also for all our audience, for those that have joined us out of interest, for those that support the foundation and support the work that Louise and her team are doing. We just want to say a huge thank you, and we look forward to seeing you online in the meantime, but hopefully meeting some of you in person in the near future.
Thank you.
Thank you very much.
Thank you.